This lab activity will investigate the molecular properties of enzymes. Enzymes are cellular proteins that act as biological catalysts. Enzymes act upon substrates to perform a particular reaction. A substrate is a molecule that an enzyme binds to during a reaction. The substrate is usually the molecule in a reaction that is being changed. A biological catalyst is a molecule that speeds up the reaction rate, but is not used up in the reaction, meaning that an enzyme can perform the same reaction multiple times because it is not used up in a reaction. After an enzyme binds and reacts with a substrate, it can release that substrate, which is usually now called a product, and bind to another substrate to perform the reaction again. Enzymes promote specific reactions, and the reaction that an enzyme participates in is determined by the structure of the enzyme. The three-dimensional shape of an enzyme determines what substrates it can bind to. Enzymes bind to substrates through their active site. An active site has a specific shape and properties that perfectly allow the enzyme to bind to that substrate. Substrate binding follows two models in the cell. The first is the lock and key model, where the substrate fits the enzyme's active site perfectly, as shown here. Notice how the shape of the substrate and the shape of the enzyme's active site are a perfect fit. The second model is the induced fit model, where the active site does not fit the substrate structure perfectly until the substrate comes into close contact with the enzyme. The induced fit model can also occur when a secondary molecule, often called a cofactor, binds to a site other than the active site on the enzyme to cause the enzyme active site shape to change shape to allow substrate binding. In this situation, notice how the active site is not a perfect fit, but when the substrate comes close together, it can interact with the enzyme structure and it induces a perfect fit. An enzyme shape is determined by four levels of structure organization. The first level is called the primary structure, which is the linear sequence of amino acids, which are the building blocks of a protein. These amino acids have different chemical properties, such as being positively or negatively charged. The different chemical properties of the primary structure allows amino acids within a sequence to interact with each other. This interaction produces secondary structure. Um, amino acids here, amino acid interactions here can make structures including alpha helices and beta sheets. When these secondary structures interact, they form higher structures called tertiary structures, where multiple helices or beta sheets can interact to form advanced structure. Finally, quaternary structure can occur when two or more individual proteins are required to come together to form a final functional protein. Not all proteins will have a quaternary structure. Enzyme structure will be covered more in detail in next week's lab lecture, but for now, it is just important to remember that a protein structure determines the protein's function. In this lab, the enzyme we are investigating is catalase, one of the most common enzymes present in aerobic organisms. This is because cellular respiration produces hydrogen peroxide as a toxic byproduct, and the cell needs to remove hydrogen peroxide to avoid oxidative damage. Catalase breaks down hydrogen peroxide into water and molecular oxygen, both of which are useful molecules to the cell. In this experiment, we will use the floating paper disc method described both in the lab manual and lab tutorial video to measure catalase activity. Essentially, the catalase enzyme on the filter paper disc will be placed in a beaker of hydrogen peroxide. The catalase on the disc will break down the peroxide into water and oxygen. The oxygen produced will form bubbles that will cause the disc to rise. The amount of oxygen produced in this reaction will be directly proportional to the rate of reaction. When the paper disc rises quickly, this indicates a fast rate of reaction. If the paper disc fails to rise, the reaction has been disrupted and will not occur. It's very important to remember that enzyme reaction rates are directly proportional to the concentration of an enzyme in a solution. For example, 10 units of catalase will perform this reaction twice as fast as 5 units of catalase will. This feature of reaction rates will allow us to determine the answer to the question of the week, which is to determine the number of catalase units in 1 gram of green pepper tissue and 1 gram of yellow pepper tissue. We will measure the rates of reactions of catalase standards to construct a standard curve to determine how much catalase is in each of these tissue samples. As some background information, green peppers and yellow peppers are the same plant at different stages of development. Peppers are green when they are younger and unripened, and turn more yellow as they become ripe and older. With this information, you should make a hypothesis for this week's experiment, which you will present in your introduction in your lab report. Remember that a hypothesis is an educated prediction of the outcome of an experiment. Hypotheses should not be random guesses. Rather, they should be made with the help of previous research. So for this week, it will be important for you to locate, find, and read primary literature sources that investigate protein levels of plant tissue at different developmental stages in order to make your hypothesis. It's always important to give your hypothesis support. In this lab, although you are not performing the lab yourself, you will be expected to write a detailed materials and methods. So please read the lab manual procedure to get a good idea of how this experiment is performed. It is important that gloves are worn during this experiment so that you do not directly touch the paper discs, as the oil on skin can affect the results. To further prevent this, the paper discs will be handled with forceps. And those forceps are rinsed in water between trials to prevent cross-contamination. All experimental conditions and standard conditions will be tested 3 times. This will allow us to make sure that our results are consistent and repeatable. If the paper disc does not rise after 3 minutes, which is 180 seconds, we will assume that no reaction will occur. No reactions will occur when enzyme function has been completely disrupted. In your lab report, you will need to state that a reaction of 180 reciprocal seconds indicates no reaction. The data given to you in your lesson page for this experiment will be reported in seconds. You will need to convert all of this data into reciprocal seconds for your graphs and tables. This will allow you to calculate the rate of reaction, which is more applicable to this experiment. To calculate reciprocal seconds, it is simply 1 divided by seconds. Please see the lab manual for more details. This lab will begin with measuring experimental standards, which will be made up of 0 units of catalase per milliliter, 20 units of catalase per milliliter, 200 units of catalase per milliliter, and 400 units of catalase per milliliter. Based upon what we know about enzymes and concentration, we should expect the 400 unit milliliter standard to react the fastest, while the 0 unit per milliliter is our negative control, which we would expect to not react at all because there is no catalase in this reaction. Remember, catalase is the enzyme that is driving this reaction. A negative control is very important for every experiment because if we were to observe the 0 unit per milliliter catalase reaction disk rising, we would know that there is something wrong in the experiment because we expect no reaction. This could be due to contamination of our solutions, mislabeling, or something else. The two experimental samples investigated in this experiment will be green pepper extract and yellow pepper extract. According to the lab manual, the extracts will be made by blending 10 grams of tissue with 100 milliliters of water for a final concentration of 0.1 grams of tissue per milliliter. This is outlined in the lab manual, and it will be very important to calculate your final answers. The experimental standards in this experiment are created by diluting a 400 unit of catalase per milliliter stock solution with dilution buffer. To make the 0 unit per mil standard, you would add 1,000 microliters of dilution buffer with 0 microliters of catalase. Oops. We will be using the equation C1V1 for concentration 1, volume 1 equals concentration 2, volume 2 to make these dilutions. The experimental procedures are outlined in the lab manual and the lab tutorial video. Please email your TA if you have any questions regarding this experiment. To write the lab report for this experiment, the lab report must follow the general guidelines for a BSc 197 lab report. Please see the lab report tutorial on ReggieNet for a review. This particular report will require one standard curve, one bar graph, and 2 data tables in your results section. All of your data must be reported in reciprocal seconds, and when producing graphs, remember that you only want to graph the average value of each data set. Standard deviation and standard error should be calculated for all data sets. There is no need for a t-test in this experiment. The standard curve produced should include error bars, the correlation coefficient, which is the r-squared value, and the slope of the equation of the trend line on each graph. The bar graph should include error bars, as well as a title and axes labels. Please see the standard curve tutorial if you need a refresher on how to make a standard curve. You'll be expected to add error bars on your standard curve and bar graph. Please see the graphing in Excel tutorial on ReggieNet for a review on how to add and customize your error bars. For this lab report, your introduction should include background information about catalase. You will need to cite your sources. Remember, these sources should be from primary literature or the lab manual. Some examples of background information that is relevant to this experiment is: why is catalase important? What does it do in the cell? What reaction does it promote? What kind of protein is it? These are just some examples. Your introduction should also introduce the experimental concept for the report. What are we observing in this experiment? How will we quantify our data to draw conclusions? And finally, what is your hypothesis for the experiment and why? This hypothesis should be clearly stated. Your materials and methods is your instruction manual on how to replicate the experiment for the reader. This should be detailed enough that you can repeat the experiment just by reading this section. There should be no bullet points or lists. This should be in paragraph form. The results section is where all of your data is reported. Remember that the data for this experiment is provided to you in your lesson for this lab. You will need to write a paragraph that reports the data you've collected, in addition to reporting your data in tables and graph. The paragraph should reference each table and graph when reporting your data. For example, An excerpt from a lab report could state the rate of reaction was measured for each experimental standard. In parentheses, we've shown which standards are used. They were performed in triplicate and reported in reciprocal seconds. And then we tell the reader where to find this data. It is in Table 1. Your data table for your experimental standards should look something like this. Remember, when you are constructing a data table, you should have a table legend, which would be above your table. In your results section, you will also have one standard curve, one bar graph, and two data tables. The one standard curve is made from your experimental standard data. The experimental samples, the green pepper and yellow pepper extracts, should not be in this standard curve because we do not know what their concentration of catalase is. The data should be in reciprocal seconds. If you have any questions on this, please email your TA. An example of a standard curve is here, where we showed the equation of the line, the correlation coefficient, a trend line, and individual data points with error bars. In your discussion section of the lab report, this is where you will explain your data. Does your data make sense based upon what you know about enzymes? This is also where you will state whether your hypothesis is either supported or rejected based on your data. It is fine to reject a hypothesis. You will not get any points off if your hypothesis was wrong. You simply need to explain your data in your discussion. Finally, the conclusion is a brief summary of the takeaway message of your report. What did we learn in this experiment? For a detailed overview of a sample lab report, please see sample lab report in the introduction lesson. This concludes the Lab 1 activity on properties of enzymes. As always, if you have any questions at any point, please email your TA or attend your Zoom office hours section.